which is just based on antimicrobial resistance. We are going to take protein, which is involved in antimicrobial resistance. So day first, that is today, we are going to um, do protein and ligand preparation. And for day two, we will do actual docking. And day three, we will perform visualization. So as we all know about lock and key model. So for every enzyme, there is a substrate which binds to its active site. So active site is important for the enzyme as it has a lot of interaction with the substrate. And also we are going to, for this session, we are going to take ligand uh, in so ligand for enzyme substrate complex. So molecular docking is a technique that predicts the preferred orientation, affinity, and interaction of ligand in the binding site of a protein. And in CADD, that is computer-aided drug discovery, macromolecules, which is protein, are generally considered rigid, and some software provides flexibility to certain residue while running docking. And the word ligand is from the Latin, which means tie or bind. So in place of substrate, we are going to use this word ligand for this session. So a ligand is an ion or molecules which donate a pair of electrons to the central metal atom or ions to form a coordination complex. And we are going to use Autodock Vina tool for docking, which is very efficient, freely available, and it comes with two components, that is Autodock tool for preparation and Vina for actual calculation. So let's start. So let's go to the site RCSB PDB by writing RCSB PDB into Google. So you will get this link, which is the first link. RCSB PDB. This is Protein Data Bank. So let's open by clicking the first link. And you will get this page. Okay, so everybody is following. Write on the chat box if you have any problem in coming to this page. So this page is that here a molecule of the month is mentioned. Every month there is different unique molecule uh, is displayed, and this data bank has this number of a structure from experimental. So these are the number of structure which are deposited in this data bank by experiments, different experimental PDB structure. And here this structure show that computed structures. So these number of structures are deposited in them by computer models. Okay. So here we are going to write about the protein which we are going to use for docking. So we have to download this protein from here, write down NDM1 since we are going to download this NDM1 and enter. So you will get this page where there is different kind of this four, this is the PDB ID, this 4-digit, that is alphanumeric. So this is unique for every protein. Now we are going to download NDM1. You can also select different like from here which release date. If you are going to, you can select for the recent like 2024, or you can also select from here refinement in resolution in angstrom like 0.5 to 1, or which kind of resolution you want and which entity you want and which experimental method you want and which bacteria or eukaryotes, or in bacteria section also scientific name of the bacteria, which like if you want it for E. coli or some other, then you can just click it over running like and running on this. You will get different kind of structure which is like NDM, different NDM structure which is from the organism E. coli. So this you can see that they are unique PDB ID. So everyone is following. If you have any problem, write on the chat box. I have asked to download Discovery Studio and I have also sent attachment in the mail. So have you all downloaded and installed Discovery Studio software? If you have, then please write on the chat box. Okay. Okay. We have downloaded. Now let's just click over here NDM1 and let's download that protein from this PDB data bank. So let's download this 5ZR8. You can also write it on this PDB ID in your this search dialog box so that you can get it directly. Otherwise, this 5ZR8 I'm going to download. So just please click on the 5ZR8. Click on the chat box, the link which is provided. So you will get on this page where this is this crystal structure of NDM1 beta lactamase. And see in this here, it is provided information like from where this has been downloaded. This organism is E. coli. Since we have selected E. coli, it has different chain A and B, and all these information about hydropathy, disorder,
सर नबीला को ज्वाइन करवा दीजिए, ऐड कर दीजिए। ओके
so we were here. Like you can know about this protein. All the information is present here in this page about the protein chain, its ID and everything. And also you can visualize this protein in 3D by just clicking it, explore. By exploring, click on explore in 3D. and then you will be able to visualize the structure, and also you can zoom it or zoom out, zoom in and zoom out the structure. So right now let's download it. Here you have this option of download file. Click on this. And for docking, we need this three-dimensional structure. So download it in PDB format, that's protein data bank format, because here it is present in three-dimensional structure. So we have downloaded this. So just double click on your download section, and where you have downloaded this, and this will be open in Discovery Studio software. So everyone is following. If you have any problem, please write on the chat box. Is there any problem? Okay, so let's move forward. So everyone is here, you have opened your protein, this 5ZR8. Or write in your chat box, or give some response if you are here at this point, so that we can proceed further.
Yes, ma'am.
Okay. One or two more students can tell so that we can proceed further. So, okay, Sana has shown this. So now let's see, just by clicking in this arrow, you can see that there is, it's not working. Which file has to be downloaded? 5ZR8 from PDB, Mahjabeen, which I have already shown. So yes, just the link which I have provided in this chat box, you can just click on the link, and I'm just telling you once more, okay? Just go on that link. So have you clicked on that link? Yes, and in the download section, just go in this download file, click on download file and download this PDB format. Okay, just click on this and your download will start. Just on this 5Z8 R8 .pdb. So is it downloaded? Just double click on this and it will be open on Discovery Studio, or you can open it by right clicking it and open with Discovery Studio. Okay. Terim, now is it open in Discovery Studio? Have you installed Discovery Studio? Terim, your file is open now, or is there still some problem? So you have downloaded that, the protein. Terim, have you downloaded this protein? Yes, okay. So you have downloaded. Go
Yes,
to
ma'am.
download. Okay. Go in the download section where you have this protein 5ZR8.pdb in folder. So it must be in your folder, right? So you can open it just by right clicking on it and open with, click on open with and then to Discovery Studio. Select Discovery Studio. Is there any problem? Terry, you have this in the download section. Just right click and then open with. You have installed Discovery Studio software, Terry? Installed properly? It's not opening. File is not opening. You are just right clicking on it and open with. Then is it not showing Discovery Studio software?
Yes, ma'am.
What? It is not showing Discovery Studio software.
Yes, ma'am. It's not showing option of opening with Discovery Studio.
So you must not have installed it properly. Uh, have you, is it showing anywhere Discovery Studio? Your download section have this Discovery Studio. Can you install it again just by clicking on it?
Do I have to install Discovery Studio again?
Yes, installed it if it is not installed properly. So when you open this 5ZR8 in Discovery Studio, it will look like this. And when you right click on this, a dialog box will appear. And in that you can see this select on mouse mode and where you can rotate it by option rotating it. Then also you can do zoom it again by clicking on the mouse mode and zoom. Zoom it, zoom in and zoom out. You can also, you can do a lot of things, other thing also. We will discuss that later on, but let's now focus on preparation of protein. So this protein, this is 5ZR8 contain different chain like A, E. So let's see what is there. A chain have this glycine at 42. So this all is amino acid residue, means they are protein. Okay. A chain, and what is this E chain? E chain has also this amino acid residues. Then A chain again has zinc because our this active site of NDM contains zinc atom, so it has the zinc atom also. So, and it also has this certain ligand molecule, which is this highlighting. When we are clicking on it, it gets highlighted, as you can see over here. Let's see this. Yes, it is also highlighted. So this is for A chain. Okay, now let's see this E chain also has the zinc atom. Now A chain also contain this water. HOH is what? H2O formula is of water. So now we have to prepare for docking. So we have to remove this water molecule. So let's click on this. Then all your water molecule around A chain is selected. As you can see, it is highlighted. Then you have to press this delete. Then this is all deleted. Now similarly for the chain E, you can see that again there is water molecule. Just select on this. It is highlighted and then press delete. Then it is deleted. Now your protein is like clean. So everyone is following till here. You have done this much. If you have any problem, write on the chat box. Um, you have opened this this protein in Discovery Studio, Mahajbeen. Okay, so you must have this kind of visualization or okay, so there is an arrow. You can click on that and then you will have this. So have you done? Okay. So now delete water molecule from the chain A and E. So have you deleted the water molecule? Okay, I'm repeating it again. So from this here, you can undo everything. Okay, just by clicking on this. So this is the protein which have different chains like A, E, and in A chain there is zinc ion and certain other molecules also, which is ligand atom, which is attached to this protein. Okay. And similarly with the chain E also there is zinc ion which is attached to this active site, which is present at the active site of this NDM. And there is also this water molecule which is surrounding these proteins. So once you click on this A chain, which has this water molecule, and then press delete, it is deleted. And then similarly, you have to do this with the chain E. Then select on chain E, this water molecules are highlighted, which is around the protein, and press delete and it will get deleted. So have you done? Okay, Karine, you have done. Majbeen, have you done? And other student, what are the problem which you are facing? You can unmute and tell me. Okay, I'm repeating it again. So whatever I have done, I can do undo by just clicking on this. Okay. So this, you must have got this display box here, right? And if you're not getting this, you can just click on this arrow, which is present over here. You will get this. Okay. Then there is also arrow with each chain. Like if you have this only simple protein, you can just click on this arrow and then you will, all the chain will be shown. Okay. And for every chain, you will click on this arrow and all the amino acid residue will be shown to you. Okay. Similarly with the chain E also you can see similar. So there is this chain A with the amino acid, and chain A also have the zinc ion and certain other ligand atom. Chain E has zinc ion and chain A has water molecule also. This is HOH is water molecule. So you will just click on it. Then you can see that all the water molecules around this chain is highlighted with the yellow color. And you can delete these water molecule by pressing delete in your this keyboard. And similarly do with this chain E also. Click on chain E. All the water molecule will get highlighted and then press delete. It will all get deleted. So have you done? Okay. So now we can see that let's delete this chain E also by selecting it. We are going to use this only single chain, that is chain A, and chain E part delete chain E. Okay. Select both and press on the keyboard delete and it will get deleted. Then we are left with only chain A. There is also one other zinc ion, I think. Yes. So this, if we have to delete the zinc ion which is from the chain E. Again, select this and delete. And let's rotate this chain. There is one more zinc ion which is not present at the active site. This one. So these zinc ion are of protein chain E. So that is why we are deleting it. We are only going to use chain A for this purpose. And chain A, you can see these two zinc ion are of chain A, while this is zinc ion is of different chain. So we are going to delete that also. So since when you click on this ion, it will get highlighted zinc 303. So we are going to delete this, and also we are going to delete all the ligand atom like these two. Let's click on this and it is highlighted over here. Again, select on this and delete. Since we are preparing protein or cleaning our protein so that we can dock our ligand molecule, that is why we are deleting all these het atom and water molecules so that our ligand can efficiently bind with the active site of the enzymes. Now let's select this also and delete. So everyone is following? And we are not deleting these two zinc ion because we already know that our this protein, which is NDM1, it has zinc ion at the active site residue, which is involved in hydrolysis of antibiotic. So we are not going to delete this because This is a part of this chain A, as we can see in this. Okay. If you are encountering any kind of problem, just write it in your chat box. We do not have to delete all zinc. If you have deleted, you can just click on this. This is for doing undo. So click on this and then select the desired zinc which you have to delete, okay? And delete those. You can undo from here. Just by clicking on this, you can undo and redo from this. So have you done? Okay. Okay. Let me rotate it. There is one ligand atom which you can see over here, which is binding to this chain, while this one is of different chain. So let's select this and delete it. And do not delete this chain, this ligand molecule, which is involved in interaction with this atom. So you can come to tool section, click on this view interaction. So you will get this two view interaction and define and edit binding site. So once you click this define and edit binding site, just firstly select this ligand molecule. And then you can define and edit your binding site from current selection. So this sphere will be shown, that is SBD site sphere, as you can see with this. So this sphere is covering the ligand. And if your ligand size is more, you can expand it. And if your ligand size is small, you can contract it. Okay. So is there any problem? Everyone is following? Okay. So when you will, okay, wait, I'm just deleting this. So firstly, what you have to do, you have to select the ligand molecule. This is SO4306. I have selected it. It will get into yellow color. It will get highlighted. I'm repeating it again. So you have this SO4. Okay. So here you will be this tool dialog box is there. Small tool is written here. So just click on this tool and you will get this two, like there will be view interaction and define and edit binding site. So both Tehreen and Majubin, are you following? Have you got this? if you have any problem, you can tools not present. Have tools not present? Why receptor? You have clicked on receptor ligand interaction here. Yeah, you have find tool, then just click on that. And then you will get this view interaction and define and edit binding site. Tahreem and Mahjabeen. So have you got this? Okay, Karim. What about Mahjabeen? Yes. Okay, then you have to click on this define and edit binding site. Okay, just click on this. And in that you have to click on from current selection, this. Okay. So just click on this from current selection and you will get this sphere once you click on it. Okay. So I was telling that this sphere you can just by click on this sphere and you can expand it and contract it both. Expand it so that if your ligand molecule size is large, then you can expand it. And if it's small, then you can contract it also. So this sphere is so this sphere is so just click on this SBD site sphere and right click on it, and then you will find this attributes of SBD site sphere. Just click on it, then you will get this X, Y, and Z coordinate of this. Then copy these coordinate for X, Y, and Z axis and paste it in your notepad. This we will use for our upcoming session. So you have to copy this. Is there any problem? So everybody has found this X, Y, and Z coordinate. I didn't get this SBD side attribute box. Have you select this SBD site sphere? Mahjabeen, you have to select on this SBD site sphere and right click on it. And then you will, in the last, there is that attributes of SBD site sphere. So you have to click on that. Then you will get this box. Mahjabeen, have you done? So you have to save these coordinates, and I will ask these coordinates in the next session, because we are going, once we will have to set our grid box, we will use these coordinates. So let's copy it and paste in your notepad. So I have saved this in my notepad. So have you saved? And then if you have saved this, let's close it. And then save this. Then you can delete this PDB site is fair just by clicking on it, selecting it and delete option. Okay. Because we were only concerned about this X, Y, and Z coordinate of this ligand molecule. So we have now deleted it. Now let's delete this ligand molecule as well because we are going to dock our new molecule, which will bind to this, like this is fair. So have you done till here? Have you deleted this SBD site is fair and also delete this ligand molecule because here we are going to dock our new ligand molecule. So once you are done, so let's save this. Now save as. In PDB format, you will get this protein data bank format, okay? Okay. Now I have this workshop folder which I'm going to save over here. This my protein. I've saved it. So now let's go and prepare ligand. I was saving, right? You can save as from here from the file section. You will get, just click on file, you will get the save as. Click on it and then save it in Discovery this protein data bank file rather than Discovery Studio file. Click on this protein data bank file and save. Tareem, have you done? So let's now prepare for ligand molecule also. If you are done, let me know so that we can prepare ligand molecule. Okay, so you're done. So now go on Google and type PubChem. PubChem. And enter. So you will get this first link. Click over here. Then write, let's talk it with meropenem antibiotic. So type meropenem. Then enter. So everyone has reached here and typed meropenem. Let me know in the chat box. Okay, so enter it. then you will get this, and then click on here meropenem. Okay, just click on it. And you will get this meropenem page. This is PubChem database where there is a lot of molecules present. So there is all the information about this meropenem antibiotic, about its molecular formula and description is there. Its 2D structure is available and its 3D structure is also available. And some more information is present over here. So you can just read about this later on, about this antibiotic. So let's download its 3D structure. When you just come to 3D conformer, and then here there is download coordinates. Click on download coordinates, and then you will get here this SDF, right? So just click on this SDF and in the save box, and then save it. As I have told you about this PDB ID, there is also a CID of this every molecule, which is present unique ID. This is PubChem CID, that is compound ID. It is like this for meropenem, it is 441130. This is also unique for every molecule. So we have downloaded its 3D format. So everyone has downloaded this 3D format or not? If you are encountering any problem, just let me know. Okay, so let's open that molecule. So that must be in your download folder. So just open it with Discovery Studio again. So let's open Discovery Studio. So you will get this. So is this visible to everyone in your own system? Okay. So since we have downloaded 3D Conformer, so you have to click on arrow here. Just click on this and you will get its three-dimensional structure. Is everyone following? Okay. So this is an SDF format. Now we have to save this ligand molecule. This molecule we are going to dock with that protein, which is NDM1, which we have prepared. So now for preparation of ligand, we have to save it in PDB format. So again, you save that in PDB format, and again in the same like workshop, and rename it as ligand. So you have saved this in PDB format. Then now for today's session, we have to, as we have discussed, that we have to prepare protein and we have to prepare ligand molecule. So we have prepared this both protein and ligand molecule. Now you have to download AutoDock Vina for tomorrow's session. So I'm just telling you how you can download that. type on Google so type on Google Autodoc Veena. Okay. Wait. Autodoc Veena. Okay. Download, click on Autodoc Veena download, and you will get this downloads. Download it according to your operating system. You will get this. So for Windows, you have to download this, or if you have this any other operating system, you can download it according to your operating system. So just click on link and download this. And you have to download one more, that is MGL2 for visualization, because before without it you cannot it will not be visible. Your Autodoc tool is not visible. So download on MGL2. Click on Center of Computational Structural Biology. Just download MGL2, and you can download MGL2 according to your operating system. So tomorrow we will do docking. So that's it for today. If you have any question, you can ask me. And I have told you this to download both the Autodoc Veena as well as MGL2. So please download it and install it before tomorrow's session. So is there any problem to anyone? If you have any question, you can unmute yourself and ask any question. So is it clear to everyone? So please, before tomorrow's session, download these two software, that is Autodoc Veena, and for visualization of that Autodoc Veena, you have to download MGL2. So please download this both and install it. So tomorrow we will be proceeding for further docking process, because we have prepared our protein and ligand molecule. Yes, there is no criteria. You can use any chain, since we are using a chain, because there is only one chain which we have to use. So we have downloaded one and used one. You can use any chain depending on like whatever you want. It's not any criteria, because both the chain were similar. So maybe I can show you if I have that structure. Wait, let me show you this 5. Yes, let me undo it. And let's, this was, no. Let me show you that protein which we have downloaded, and its chain A and E. Yeah. So see, this chain has this glycine, aspartate, glutamine, and similarly you will find that chain E has also at the position 42 glycine, aspartate, glutamine. So these both chain are similar. So that is the reason we have deleted one chain and used one chain. So if your chain is not similar, you have not to delete any chain. Okay, so that for that case, you have to use all chain if your chain is similar, then you can only delete one of the chain. Okay? Is there any other question? Is there any question you can ask in the next 1 minute? Otherwise, we are going to close the session. Okay? So you have 1 minute to ask the question. So no questions? Hope you have enjoyed today's session.
Okay, thank you, Nabila. If the session is completed, then let me close the meeting if there's no further question. I think tomorrow's session, if they have any question, they can ask on tomorrow's session. Okay. Okay.
Okay.